Hello and welcome to my presentation towards in-silico drug design. My name is Marta Stępniewska-Dziubińska. I work as a software engineer at NVIDIA, and my job is to optimize deep learning models for drug discovery. Before I jump into in-silico part, let me first briefly talk about drug discovery in general, how this process looks, how we find new drugs. So we start by analyzing molecular mechanism of the disease. We need to understand how the disease works in order to cure it or treat it. When we understand this mechanism, we select the target. It's usually a protein that plays an important role in this mechanism, and we want to attack this protein. Then we find small molecules that can interact with this protein and modify its activity, for example, disable it, leading to a therapeutic effect for the patient. And we're done, right? We have our new drug, end of the story. Of course not, it's not that simple. First of all, how do we even find such small molecules that can interact with a protein? Secondly, it's only the beginning. So after drug discovery phase, there's also drug development phase, during which we make sure that our molecules work for the patient as expected. So when we give it to the patient, they have this therapeutic effect, but at the same time, then they don't have any severe adverse effects. This entire process, drug discovery and development, is very costly and very long. What is worse, the success rate is very low. Around 80–90% of the molecules that we design during this drug discovery phase will fail in clinical trials. So, for example, they will turn out to be toxic for the patient. So the idea of in silico methods is to make this process more efficient. We want to be able to focus on the most promising small molecules during drug discovery phase, and we want to predict which ones of them will fail in clinical trials because of the, for example, toxicity. In silico drug design is a cycle during which we iteratively improve our molecules. We start from getting the structure of the protein target and building the database of small molecules, of our candidate molecules. We can do it either by pre-filtering some larger existing database of small molecules, or we can try to use de novo design to generate our molecules from scratch. When we have our protein target structure and the database of candidate molecules, we perform so-called molecular docking, which is simulating the interaction between our structure and those small molecules. And from molecular docking, we get so-called protein ligand complex. So small molecule in this context is called ligand. And we can analyze this complex to understand how our molecules can interact with this protein and to try to optimize our molecules to get stronger interaction. And this way we get an updated version of our database of candidate molecules. So let's first talk about getting the protein structure. So our goal is to have 3D coordinates for our protein of interest. This is important because 3D information gives us more insight into how this protein works and what kind of interaction it can form. Theoretically, you can do it experimentally, but it's very expensive and time-consuming. It can take years to get the structure of a protein experimentally. And also, proteins are not that easy to, for example, crystallize, which is an important step in getting the structure of the protein. So we can spend years on getting the protein structure experimentally and fail. So alternatively, what we try to do is to use protein sequence, so the chain of amino acid that form this protein, and perform in silico folding of this protein, so to predict its structure. And many of you probably already heard about AlphaFold, so I wanted to show you a different model that performs protein folding. Similarly to AlphaFold, it takes MSA as an input. So this is our protein with similar proteins aligned together, which gives our network some evolutionary context in which it can analyze our protein of interest. As another part of an input, we can provide so-called templates. So some features for proteins that are similar to ours, but we know how their structure looks. So it gives our network some structural hints about how the structure of our protein might look like. And similarly to AlphaFold 2, this network is a set of transformers that process this data and exchange information. What is different is that we also have a 3D track, which uses SE3 transformer and explicitly works on 3D information. And this is also the reason why I wanted to show you this model, because SE3 transformer was optimized by our team to make it faster and to make it use less memory to train. So this is a network that is equivalent to 3D rotations and translations, which means that if we rotate an input, we will get rotated output from this network, for example. And this is very important property for proteins because their orientation doesn't change the protein. It's the same protein if we rotate or translate it. And this model is used in two places in this network. So in this 3D track and also for the refinement of the predicted coordinates. So getting this part faster make it possible to remove two bottlenecks from this network and make this entire network faster. Okay, so we have structure of our protein. Now we can focus on the database of candidate molecules. And let's first talk about filtering our database of molecules based on the properties. So for this, we need to select molecules with desired properties and also discard those that, for example, might be toxic. But of course, it's not straightforward to do. We cannot just tell what this molecule will do in the patient's organism by looking at its structure. This relationship between properties of a molecule and its composition is not trivial. So what we can do is to train a deep learning model that will take such molecule and predict its properties. Then we can apply this model to large database of molecules and use the results for filtering. And an example of such model is ERMAT. It was developed in collaboration between Jagiellonian University and NVIDIA. And this is a graph transformer that works on small molecules. So as an input, it takes 3D molecular structure, which is also something unique for molecules because we have a graph in which each node has 3D coordinates. And this model uses a very clever way to represent relationship between nodes in the graph, and it makes it easier for the network to understand how basically chemistry works. So we represent here distance in the graph between the nodes. We represent the type of bonds that are between the atoms in our molecule, but also we represent distances, but not as a single number, but rather as an encoding, which makes it easier for the network to learn some nonlinear relationships. Okay, so we are working on our database of molecules. We used filtering of a larger database to get our molecule candidates, but we can also try to generate our molecules from scratch. And our goal here is to get a database of molecules that already has desired properties. We cannot do it by just exploring the chemical space at random or systematically because it's too vast. There are simply too many possible molecules to just generate all of them and check their properties. So what we can do instead is to use a deep learning model with well-organized latent space for molecules and sample from this latent space. So we often use a seed molecule as a starting point and, for example, generate molecules nearby in the latent space. Some models also provide an option for optimizing our molecules based on selected properties. So they take as an input also some property constraints for the molecules. Here you can see, for example, that we would like to get a molecule similar to the one presented on the left, but less hydrophobic. Log P is a measurement of how hydrophobic the molecule is, and we want this measurement to be lower. But we don't want to change this molecule too much, and we would like to keep other properties roughly unchanged. So we can ask our model to keep our other properties unchanged and modify log P a little bit. And we end up with a very similar molecule. There is only one chemical group difference between them, but the log P is lower. And an example of a model that can do such things is MegaMolBERT. This model was developed in collaboration between AstraZeneca and NVIDIA. And this is a sequence-to-sequence transformer that was inspired by natural language modeling. This is basically BART model adapted for molecules. But wait a second, I just mentioned that molecules are 3D graphs. So why am I showing you a text-to-text transformer? This is possible because there is something called SMILES. This is a molecule representation that allows us to encode the molecule as a string. And you can see some examples of such strings presented here in the diagram. So this way we can adjust the models that were designed for language modeling to work on molecules. And what was very clever in training of this model is that they use a property that is unique to SMILES. You don't have it in natural language. And this is, I think that SMILES are not unique. So we are able to create multiple SMILES strings for the same molecule. And then we can train such autoregressive model by providing it with one SMILES and asking it to predict another SMILES representing the same molecule. This model was trained on a very large database of molecules. The original version called Conformer was trained on 100 million molecules, and the MegaMolBERT version was trained on 500 million molecules. So this model knows a lot about chemistry and has really nice latent space for generating new molecules. Okay, so we already have our structure of a protein. We already have database of candidate molecules. Now we can perform molecular docking. So our goal here is to understand how and if the small molecule can interact with a protein target. And the challenge here is that proteins have multiple regions that can form interactions with other molecules, so-called binding sites. And we don't know which one will be used to form this interaction. Often we don't know where those binding sites are located. Another thing is that small molecules are highly flexible and their conformation changes when they form protein-ligand complex. So again, we have too many possibilities to explore systematically. And what we can do instead is to train a deep learning model that will take protein structure, that will take small molecule, and will perform docking for us to get the complex. And an example of such model is EquiBind. This model works on 3D structures for protein and for ligand, and they are represented as so-called spatial KNN graphs, which means that nodes in the graph are connected to K nearest neighbors. So not necessarily to the other atoms or other amino acids that are connected to our node with a bond. What this model does is predicting key points. So we have two sets of key points for each of the molecules that correspond to each other. And if we overlay those key points, we will get the results of the docking. Another thing is that it also predicts the right conformation for the ligand. So we can feed it with the random conformation and it will change it to the right one. What is very cool about this model is that, similarly to the SE3 transformer, is equivariant to 3D rotations and translations, but also it has soft constraints on the ligand flexibility. So it is more probable for the model to modify the structure in a way that is relevant for forming the bond and keep the elements that rarely change in the conformation intact. And the coolest thing about this model is that it performs blind docking. So if we use classical tools for protein docking, for molecular docking, we have to first either find binding sites or on the receptor surface with another tool, or we can search this surface while docking, and we just have to try to dock our ligand everywhere. And this model in one go predicts how the ligand binds to the protein and also where. So it's less computationally expensive, and we can apply it to the larger databases. Okay, so we have our protein ligand complex. Now we can analyze it and try to make sense out of it. And our ultimate goal in drug design is to find ligands that have the strongest effect on our protein target. But docking doesn't really give us this information. Some models return kind of score that tells us how well this molecule is bound to the protein. But for example, equibind doesn't give us this information at all. So what we can do now is to use another model for predicting so-called binding affinity. So the strength of the interaction between this small molecule and our protein. And given the score, we can discard those ligands that don't interact that strongly with our protein, so they have weak affinity. And we can focus on the ones that have moderate or strong affinity. What we can also do is to look for the improvements in the structure of our small molecule to basically make it a better drug candidate and interact strongly with our protein. An example of a model that can predict binding affinity is Pavnuce. This model is already a couple of years old, but it's mine, so I wanted to show it to you. And the idea here is very simple. We take 3D protein ligand complex, we calculate features for atoms in this complex, both for protein and for the ligand, and we put it in a 3D grid. Then we pass this grid through a 3D convnet, and this convnet predicts binding affinity. So it looks like old models for image classification, but it works in 3D. And as I said, this idea is very simple. Also, we don't take into consideration some domain-specific knowledge, like, for example, the fact that rotating protein ligand complex doesn't change its properties, but it works surprisingly well. And we can get pretty good binding affinity estimates with this network. Another convolutional neural network that does something different is DeepRag. It also analyzes protein ligand complex as a grid of features, but first difference is that it uses separate sets of features for the protein and for the ligand. And the second difference is that it doesn't predict binding affinity, but rather it predicts how we can improve our ligand to have the stronger interaction. It predicts fragment that can be added to our ligand to form, for example, some additional contacts with the protein. And this network is trained by removing part of the ligand. So we have our real molecules that we know that interact strongly with a protein. We remove part of it, and the network's job is to reconstruct it. And this way, the network learns how it can improve interactions between ligand and protein. Okay, so we did the full cycle. We first predicted the structure of our protein. We designed a database of candidate molecules by prefiltering some larger database based on the properties, or generating our molecules de novo. Then we perform molecular docking for each molecule in our database. We get protein ligand complex, and we analyze this complex, discarded ligands that didn't interact strongly with the protein, and look for improvements for the ones that were okay or were very good. And this way, after getting those improvements, we have a new updated database of candidate molecules. Now what we can, for example, do is to generate new molecules that are close in the latent space of our de novo model, new molecules similar to the ones that we selected from protein ligand complexes. Or we can use some network that will hint us how we can change our ligands to get stronger interaction. If we have this new database of molecules, we should probably use some property prediction model to make sure that new molecules that we proposed are, for example, not toxic and that they will work for the patient as expected. So to wrap it up, we were talking about drug discovery. but it also affects drug development phase. So by using in silico methods, we learn more about our protein target. Then we can use those methods to anticipate and mitigate problems that can occur in clinical trials. And also, they allow us to predict properties of molecules and therefore focus on the ones with the best properties. And this translates to smaller costs and faster development of new drugs, of course, assuming that we did everything correctly. So it was a very high-level overview. If you would like to learn more about any of the models I presented, here are some resources to the papers and to the code. Also, I encourage you to take a look at our deep learning examples repository, where you can find SE3 transformer implemented in PyTorch. I mentioned this model in the context of protein folding, but it can also be used for property prediction if we equip it with a different head. And you will find such example in our repository. There is a README that will tell you how to build the Docker image, how to run this model, and how to train it, what results you can expect from it. And if you find this topic fascinating, as we do, and would like to, for example, join our team, just drop me an email at martas@nvidia.com, and I will be happy to answer all of your questions. Thanks.
Oh, thank you for a very interesting presentation. So we have a question from the audience. What is your opinion about Google AlphaFold AI? Does it work as it is supposed to do?
Okay, so I tested it on a very limited sample of data, and it works really good. It works really well. It's on par with the experimental results. The thing that is unclear for me is how it would work on proteins that you cannot really crystallize. So I mentioned that it's not always possible, because it definitely learned to do the task that we already can do experimentally. And it's not really proven that it's good at solving structures for different kinds of proteins. For example, I would be interested to see more information about using it for membrane proteins that are very, very different from the ones that are widely available in the databases. So yes, this model is really revolutionary. This is, like obvious thing by looking at the results. It's on par with the experimental results for the model, for the proteins that we can solve experimentally.
Oh, thank you. So I would also have a question about your model, because you mentioned it's not, it is sensitive to rotations. On the other hand, you have already found a solution to make a model insensitive to rotations that was applied in earlier models. So do you think it will be possible to somehow reuse this solution and to expand your model, or perhaps, because you also mentioned that it works good despite this limitation? So
yes, so I think the thing that can be reused from Pafnuce is the way I encode features. So I think it really works well, the way we encode those features, and how we can improve it by changing the network architecture. So this is 3D convolutional neural network. And for example, if we use graph convolution for this, or if we use, for example, SE3 transformer, I bet that we can get better results. But this featureization that we have for Pafnuce is very powerful, and I believe that it can be reused with more up-to-date, more recent models.
Oh, thank you. And maybe one more question from my side. In the abstract, you mentioned that some models coming from different domains may be reused and repurposed here for drug discovery. So could you briefly comment on this? So was any of this, the model presented during your talk, was it reused from other domain, or they were all
yes? So MegaMolBART is a model that I think we can call being reused. Also, Pafnuce was very much inspired by LeNet-like networks for image classification. So the idea here is to look how we can represent biological data in a way that they mimic data from different domains. And for MegaMolBART, we are able to represent small molecules as text and then use text-based models to analyze them. And for Pafnutse, it was the idea to represent protein as an image. So we pixelize this protein to make it work like an image and analyze it with a network that can work on 3D images. And this was because a couple of years ago, there were not that many models that were domain specific for bio and cheminformatics. As I mentioned, this equivariance to 3D rotations and translations was very heavily researched, but it wasn't really a thing back then. And that's why people try to use different models, different approaches, because other domains were more, let's say, mature in deep learning, right? So now I see a very big boom in this equivariant models that are very domain specific, and I believe that we'll get way better results with them than with those models adapted from different domains. But again, it gave us a good starting point because we, for example, know really well how to train transformers effectively, right? So we can, if we're able to represent something as a text, we can build on this transformer language-based knowledge to have a good model for our domain.
Mm-hmm. Yeah, indeed. Thank you. And the last question from the audience, did you try any approach on raw data without feature computation?
It's not that easy because what is a raw data for chemical molecules, right? We have atoms and we have to describe those atoms somehow. So we have atom type and it can be considered raw data. We have, for example, partial charge, and the things that are used as features are basically ways to encode this raw information in a computer understandable way, right? So some of those things can be considered raw data. Also, I would say that we can say that, for example, Rosetta Fold that I presented at the very beginning, it in a sense works on raw data. So of course, we require some encoding to represent this raw data, but MSA is a very basic source of information. Older models use different approaches to analyze those MSAs and pass such features into the network. So to, let's say, extract this evolutionary context that I mentioned. So to sum it up, it's not really possible to use raw data because those things that we try to represent are not like numbers, right? We cannot just represent them this way. So we have to use some features to encode this data. But here there are different levels of featurizing data. And I believe that using these simple features give better results than some sophisticated features.
Oh, I see. Thank you. So thank you once again for a very interesting presentation and for all this explanation. It was really, really interesting. Thank you.
Thank you.
And